You start with acetyl-CoA up top with its 2 carbons, okay? And I need to back this up so I can make a big old circle. And then you come in like such, okay? So you have substrate and you have a product, right? So over here you have a 4-carbon compound. Over here you have a 6-carbon compound, okay? These have fancy names. Actually, I can make that a little bit bigger for you. So this is oxaloacetate. Again, don't be intimidated by these names. They're just new names. Learn them. It's a new word. There you go. Oxaloacetate, 4 carbons, bonds to acetyl-CoA, 2 carbons. This makes citrate or citric acid. And this has 6 carbons. 4 plus 2, 6. Now, this kicks down to only 5 carbons. The name of that substrate isn't important. If you look them up, there's isocitrate and glutarate and alpha-ketoglutarate and fumarate. And if you get into biochem, you'll memorize those and you go, okay, and you take a test. And then in all honesty, you forget them because you never have to need them again unless you teach it. All right. However, there's an oxidation reduction. So there's an oxidation reduction and well, there's a new way to spell decarboxylation. How about if we put a C in that and an E too? Here's decarboxylation. Those 3 happen, okay? You then come in here. You get your NADH, which is going to be very important with the electron transport chain that we will talk about next. And then CO2. Okay, so you go from 6 carbons to 5. Why? Because there's a decarboxylation. And then you get oxidation of your substrate, reduction of the electron carrier. Fine. Now you're down to 5 carbons. What happens again?
Oxidation, reduction, and decarboxylation.
Yep. Simply happens a second time. Nice. Oxidation, reduction, decarboxylation. Which means you're left now with 4 carbons. Same products. NADH, CO2. See, people walk into this cold and they hear the language that we speak and they see a bunch of, or they hear a bunch of vocabulary words that they've never heard of before, and they let themselves be intimidated by unfamiliar terms. And they assume it's difficult, it's complicated, and it's not. It's just unfamiliar. And what you do is through repetition and going through it multiple times, you then start to see the pattern to what's going on. And all of a sudden, science really isn't that difficult. You have to have the patience to see something you've never seen before, not be intimidated by it, and make the vocabulary your own. It's really what effectively learning science comes down to. So, substrate level phosphorylation, there's some GTP in here. If you want that detail, great. Big picture idea, you make some ATP. Still 4 carbons. No change here, okay? You then get another oxidation reduction event. What is produced?
FADH2.
FADH2. It's the only place you'll see it. Hey, guess what? Still 4 carbons, right? You get one last oxidation reduction event. Let's see, let's do an arrow out this way. NADH. And there you go. There's your big picture idea of Krebs. Okay, 2 carbons in the top here. 1 carbon out here, 1 carbon out here. Okay. 4 plus 2 is 6. 6 minus 1 is 5. 5 minus 1 is 4, and it's 4 for the rest of the time. You have coupled reactions here. You have coupled reactions here. You then substrate level phosphorylation for 1 ATP, kind of a token ATP. The appearance of FADH2, and then one last NADH. When I ask for you to draw out Krebs on the exam, this is what I want, okay? I don't want any of you drawing this thing. You'll get into the details of this if you're a bio major when you're a junior in college. You'll see. Okay, and actually, when you get into this, it's actually not complicated. okay? Because what actually happens, you see this OH here? This hydroxyl group moves down 1 carbon. And that's how you go from citrate, ready, to isocitrate. Wow. Okay, so really, even when you get into these details, it's really not bad. Okay, so I don't want you getting bogged down by the details here. Concepts, big picture ideas. When I ask you to draw out Krebs on the exam, this is what I want. And then really what I'm after is your explanation of this. 4-carbon substrate starter, what happens to the carbons? High energy electrons in acetyl-CoA, how are they ripped out? And then you make your token ATP. Okay, so please understand that's what I'm digging for on the exam. Any questions on this before we leave it?
No.
Good. Yeah. Okay. All right. All right. So then before we get into the electron transport chain, as always, I want to be sure that you're comfortable where we are in the cell. So you look at the cell, plasma membrane. Okay. So let's get in on this. That's the plasma membrane. That makes this the cytoplasm. Okay. So I'm going to draw the mitochondrion ridiculously big because that will give me more room to work. So of course the mitochondrion is not this big. Of course it's not. Okay. This is the outer membrane of the mitochondrion. And then of course there is an inner membrane with its folds. These folds are called cristae. Okay, so this is then the inner membrane of the mitochondrion. So just like the chloroplast, the mitochondrion has a double membrane. Okay, glycolysis happens out in the cytoplasm. And then in the center in here, in the matrix, is where you have Krebs. Krebs in the matrix. Okay, so is there NADH that's made out here in the cytoplasm? Yes. Is that NADH then shuttled into the matrix? Yes. Does that consume a little ATP? Yes. Do most people account for that? No. Okay, so that's where the whole 32, 34, 36, 38 comes from. Fine. So there are other processes that consume ATP that is produced in this process, and that's why there isn't always one consistent number. Fine. So embedded in the inner mitochondrial membrane are different proteins that handle the high energy electrons and pump protons. So what do we call these clusters of proteins that handle the electrons that are ripped out of macromolecules?
Electron transport chain.
You bet. These are electron transport chains. Okay, it's a series of proteins that handle the electrons, and then in these, each of them pumps protons. It's not just one. Like in Calvin, actually all of them pump protons. There's a lot more proton pumping here. So it is an evolutionary advantage to have more folds in your mitochondrion, inner membrane. It's an evolutionary advantage to have more electron transport chains. So even evolutionary fitness even becomes part of your mitochondrial proteins, okay? And then what do you think is in addition to all of these electron transport chains that I'm drawing in red? What is this thing?
ATP synthase.
Yep, this is your ATP synthase, okay? So this is the difference between an electron transport chain and an electron transport system, okay? So a chain is just those 3 black proteins. As soon as you take into account the ATP synthase, then it's an electron transport system, okay? So if you come in here and you say all 4 of these like such, that is an electron transport system because you've taken the electron transport chain and the ATP synthase. So here's what happens. You get the electrons are actually dropped off inside of the matrix, okay? And then what happens is the protons that are here in the matrix are then pumped out like this, and the protons actually gather out here, or they're pumped here. So different from the thylakoid in the chloroplast, where the protons were pumped towards the center of the thylakoid into the core of the chloroplast. That's not what happens in the mitochondria. Here it's the opposite, all right? Here the protons are pumped from the matrix into what's called the inner mitochondrial space. Okay, so the protons are then pumped the opposite direction. Then the protons return. Where should we have them return? Oh, let's have them return over here. They then return through this ATP synthase, like such. And then this is where your ATP is made, like such. And then the protons cycle back to the matrix, at which time you eat more food, oxidize more high energy electrons. The high energy electrons are delivered to the matrix and you pump more protons. And the protons simply cycle in and out of your mitochondria. Also, something here that we don't talk about and we don't even mention in high school curriculum at all, do mitochondria have their own DNA?
Yep.
Yes, they do. Okay. In fact, when oxygen is taken into the mitochondrion, the oxygen is actually ripped apart, right? And so then you have an oxide in your mitochondria. It's highly, highly reactive and actually oxidizes the DNA in the mitochondrion, which is a major contributor, we're pretty sure of. We don't just think it, we're pretty sure of it now, that causes aging. And so this is the idea of eating a lot of fruits and green leafy vegetables that contain, have you heard of antioxidants? Right? You eat a lot of antioxidants. It then stabilizes the oxygen in your mitochondrion, prevents that free atom of oxygen from oxidizing your mitochondrial DNA and slows the aging process. Very cool. Okay, so what we're going to talk about is the electron system here. So when we draw the membrane, actually we're not going to draw it at all. When you see the membrane, when you see this membrane here of the electron transport system, the electron transport chain, along with the ATP synthase, these four are those four right there. So this membrane here, this inner mitochondrial membrane, is that inner mitochondrial membrane there. And then I think the only thing that I need to label, I should label the ATP synthase. I don't think I labeled that. ATP. Cool. Looks like checkers. That's cool. Any questions on this before we move on?
No.
Okay. All right. Do it. So, come up to the top of this guy here. On top of this, and let's draw an arch. Why would I draw that arch? What is that arch?
Plasma membrane.
I understand it's tempting to say that's the plasma membrane. Super tempting. Ooh, the outer membrane of what?
Mitochondria.
Mitochondrion. Question mark, mitochondrion? Yes. It's the outer mitochondrial membrane. Extremely tempting to consider that the plasma membrane's not. Okay. So then that means out here, what's this out here? What should we label this as?
Cytoplasm.
Cytoplasm, you bet. So as with any diagram, remember where you are in the cell, always. Otherwise, these diagrams are meaningless. So those three purple blobs there are what I drew in black here. These are the proteins of the electron transport chain. Okay, then what I drew in red here is ATP synthase. It's right there. So it's always 3 in 1. All right, so let's start here. Let's start with NADH. So NADH drops off its high energy electrons to the proteins of the electron transport chain that are embedded in this membrane. Which membrane in this? This is the inner mitochondrial membrane. Okay, meaning that this is then the core of the mitochondrion here, This is the matrix. Right? So your NADH, whether this is from glycolysis, whether this is from Link, or this is from Krebs, doesn't matter. Okay? That electron carrier shows up here and drops off its high energy electrons. Once the NADH drops off its high energy electrons, what does the NADH become? It goes right there.
NAD plus a proton.
Excellent. That's NAD plus its associated proton. Okay. So then what you get is this is the path of the electrons here. Okay? This is like the yellow brick road from Dorothy and the Wizard of Oz, right? Except this is the path that the electrons carry. Okay? And these electrons then are passed from protein to protein to protein, just like such. So you don't have to worry about the names of these proteins. There are cytochromes and there's FDs and you don't have to worry about that. Okay. Know the function of these. That there are two sub proteins, and these rock back and forth between the proton pumps, handing the electrons from proton pump 1 to proton pump 2. And there's another protein over here that rocks back and forth, transferring the electrons from proton pump 2 to proton pump 3. The name of them, who cares, right? This is conceptual understanding. I want you to know the role that these proteins play, right? So when NADH is then oxidized, right? So there's oxidation here. The NADH is oxidized, the NADH is oxidized into NAD, right? When this electron passes over this protein grabs a proton from the matrix and translocates, is the fancy word. Most people call it pumping. The proton is then moved, translocated, pumped from the matrix into the inner mitochondrial space. This is called the inner mitochondrial space. And since, perhaps I should show you, since this protein pumps a proton, right, from the matrix to the inner mitochondrial space, then this is called a proton pump. The pumping, why that verb is so important, is this protein continues functioning regardless of the number of protons that build up here. The pumping continues. That's why it's pumping and not diffusion. This is definitely not diffusion. This is pumping. The proton is forced against the concentration gradient. So this one's a proton pump, and this one is a proton pump, and the third one here, oh yeah, we can put that there. This is also a pump. So there's 3 proton pumps here instead of just the one that is present in the electron transport chain of the chloroplast. Okay. All right, well then what do you think that box is? If this one's NADH, which box is this? Which carrier is this? Totally, FADH2. FADH2 is also oxidized down into, I'm not going to insult your intelligence, into FAD and its 2 associated protons. Proton is pumped again, right? Then the electrons that came from both NADH and FADH2 are delivered to the 3rd pump. There is a final translocation, there's a final pumping event, and you now end up with more and more and more and more protons up here. So you start building what?
A proton gradient.
Yeah, we're building a proton gradient here. Don't forget, fungus do this same thing. Plants do this same thing in their mitochondria. Now, how do bacteria make ATP? What bacteria do is these pumps and this ATP synthase are actually embedded in the plasma membrane of bacteria. Okay, and so what bacteria literally do is they pump the proton out of their cells and create a mini proton gradient right outside of their cells. And then those protons then come back in through an ATP synthase embedded in their plasma membrane. Okay, so mitochondria still do cell rest. They just do it just a little bit differently because they don't have mitochondria. All right, so we now have this proton gradient that's all built up. So of course, what's going to happen, actually I should label this. This is your proton gradient here, right? You have an extremely high concentration of protons, right? You then have a lower, let this go away, there you go. You then have a lower concentration of protons in here. Okay, so of course then there's a flow. There's a flow here. What's the name of that flow of protons here?
Osmosis.
Yep, that's chemiosmosis. Okay. Osmosis. It's a flow. It's a trap. It's a flow. Okay. You then have what we refer to as oxidative phosphorylation, right? Do you remember how in the chloroplast we had photophosphorylation? Okay. We call that photophosphorylation in the chloroplast because that phosphorylation ultimately is powered by sunlight. Okay. Whereas here, the phosphorylation that happens here, how is this powered, right? Where's the energy come from? It comes from the oxidation of these electron carriers. So this over here is called oxidative phosphorylation. Oxidative, yep. Simply takes an ADP plus an inorganic phosphate and makes ATP, at which time the proton returns, then simply be recycled. And it's just this flow. And this, of course, is ATP. Synthase. You okay so far? I have a thread sticking out there. It's driving me crazy.
Yep.
I think I got it. Cool. All right. What about this? Who picks up these low energy electrons at the very end? Because they started as high energy.
Oxygen.
That is oxygen. Okay. O2. Right? So of course, we then add plus 2 protons. Right? And so then the 2 protons plus 2 electrons make 2 hydrogen. But you have 2 hydrogen. But what's the chemical formula for water?
H2O.
H2O. But oxygen is what we call a diatomic molecule. It's made of 2 atoms of oxygen. So in order to make water, what has to happen to the oxygen in order to make water? Yes, it has to be ripped apart. And then these individual oxygens may be reduced. It's a great point. I'm going to add that right there. Nice. Reduction, okay? This is where the aging process comes in, okay? Organisms that breathe oxygen, when the oxygen goes into our mitochondria, the oxygen has to be separated. Why? Because it only takes one of these oxygens to bond to the 2 electrons, 2 protons, thus 2 hydrogen to make water. So this diatomic oxygen here is separated, okay? And these are what are called free radicals. It's not my terminology. This is the chemical terminology, okay? So while these free radicals are extremely important to pick up those low energy electrons, all right, these little free radicals are incredibly reactive. and bond and interact with the mitochondrial DNA. They disrupt the function of the DNA, and they then start to disrupt the biochemistry of the mitochondrion, and your mitochondrion then don't work as well. Your mitochondrion age. That's what aging is. Aging is when your biochemical systems and thus your physical systems don't function as well as they used to. A major contributor right here. Okay, so how can you slow this biochemical destruction down, right? Fresh fruits and vegetables, okay? Because what those compounds do is they contain what we call metabolic cofactors, and they bond to these free radicals and stabilize them. They're still available to be reduced into water. They're not as destructive to your mitochondrial DNA. And this is why people who eat more healthy, they eat more fruits and vegetables, age more slowly and are healthier. It comes down to chemistry. It's simple. Okay, so sometimes you'll watch, I don't know, these blenders. What's the Magic Bullet? Have you heard about this? It's this really powerful small blender that I see on TV all the time. And they will talk about grinding fruits and vegetables, especially kale, little GMO, and they will talk about needing to bind your free radicals and slow the aging process. And it sounds like a gimmick, and actually their chemistry is spot on, but they say it in such a salesman business type way that it sounds gimmicky. And I sit back and go, actually that chemistry is accurate. I wish they said it more seriously and people would take it more seriously. Let me think. Oxidative phosphorylation, oxidation, reduction, the pumping, the free radicals, the matrix. I think that's it. Any questions on this before we leave it?
No.
No? Okay. Imagine if I'd showed that to you a year ago and said, we'll be able to cover this in about 12 minutes. Okay, it's because of all your background, right? That's how we're able to do this. All right, one last flow chart and we're done. You sure? No questions? I'm that good?
Yes.
Yeah, okay, good answer. All right, fine with me. All right, big picture idea. Okay, so macromolecules. We're going to talk about proteins and how we metabolize proteins. We're going to talk about carbohydrates. And we're going to talk about fats. One form of lipid. Not going to hit the oils and waxes because they end up other places. So proteins. Proteins are then broken down into amino acids. Okay. Think of the structure of an amino acid. Amino acid has that central carbon. Then what? The R group. What else?
Amino group.
An amino group, excellent.
And then acetyl group.
And an acetyl group. What's going to be an acetyl group, right? What do you notice about this right here? It's a 2-carbon compound, yes?
Yes.
You know about a 2-carbon compound that would drop right in here very, very easily? Very easily?
Acetyl-CoA.
Acetyl-CoA. Okay, when we eat protein, right? When we eat protein, this part of the molecule here is very useful to us. Depending on what the R group is, there's useful carbons over here. But this 2-carbon backbone right here is ripped out, and depending on the R group, all right, this amino acid can be dropped here into glycolysis, can be dropped into link, can be dropped into Krebs, okay? But that's the 2-carbon backbone, okay? What do we not have a use for?
The amino group.
Yep. You're right, the amino group, which we then tag a hydrogen to and we get rid of. And the color of this compound is yellow. So where does this amino group end up as a waste product, actually. What is
The
this?
urine.
Yep, this is the yellow of your urine. Okay, so when we urinate, right, what is released? It is the metabolic water from the end of the electron transport chain, and it's the amino group from the amino acids that we've eaten. That's really what urine is, mostly. Now, we can wash out other things depending on what is in our diet, but that's where the proteins are broken down to the amino acids, the amino group comes out, and depending on the R group, this can be dropped anywhere in here. Then you have glycolysis here. Okay, and then we talked about glucose to pyruvate. And right in the middle of this is good old glyceraldehyde 3-phosphate. Does that look familiar?
Yep.
Yeah, that's G3P, yes? So when you get into Krebs, when you get into glycolysis halfway through, that 6-carbon chain rips apart into 2 G3P. It's just Calvin backwards, right? So then you come down here to the link reaction, which then forms acetyl-CoA. That should be a lowercase a. Sorry about that. That should be CoA. That should be tiny down there. So you say, but hold it. What about this? It depends on which sugar this is. It depends on whether it's a monosaccharide, a disaccharide, or a polysaccharide, then determines where, because there's actually 10 steps here. Depending on how complicated this sugar is here, determines which step this sugar then drops into glycolysis. That's why this arrow is just kind of glycolysis, because it all depends on how complex this sugar is. If it's a simple sugar, it drops in later in glycolysis. If it's a complicated sugar like a polysaccharide, then it drops into this process earlier. It all depends. All right, fats. When you look at a fat basically is made of a head and 2 or 3 tails, yes, like such. What's the head and what's the tail?
That is glycerol.
Head.
Yep, it's a glycerol head. Nice. What about that tail?
The fatty acid tail.
Nice. It's a fatty acid tail. Now, glycerol is a 3-carbon compound, okay? And that 3-carbon glycerol, you change one little side chain and you immediately convert glycerol into, guess what? Glyceraldehyde 3-phosphate. This molecule is extremely closely related to this one. It's one little change on the OH group that's bonded to the carbon, and you convert the head of fats directly into G3P. Okay. Now think about the fatty acid tail, because we all know that fat is loaded with energy, right? 1, 2, 3, 4, 5, 6. Why? Why is fat loaded with energy? I mean, we all know it is, but now you know why. What's bonded to all of those carbons?
Hydrogen.
Hydrogen. And what do you think all those hydrogens are loaded with?
What?
High energy electrons. Exactly right. This is why fat contains so much energy. It's really not the glycerol head at all. The glycerol head, you convert it into glyceraldehyde 3-phosphate. Fine. That's really not where the money is. The money is in those tails. Most tails are 14, 16, 18, 20 carbons long, right? So you've got 20 carbons, each with 2 hydrogens. You got 20 more carbon, you got 20 more carbons, right? So you got 20, 40, 60 carbons, each of them with 2 hydrogens each. These tails are just loaded with high energy electrons. That right there is why fat has so much energy in it. It's in the tails, okay? Your enzymes then simply break these long tails into groups of 2, like such. Why? Why groups of 2? Why is it always groups of 2? Exactly. those tails are converted into little pieces of 2 carbons that are immediately converted into acetyl-CoA and dropped into Krebs. Okay, this is why fat is the energy storage molecule for all mammals. This is why fat is the energy storage for, ready, all plants. Okay, it's chemistry. This all comes down to chemistry. So this is why, if you're going to be a bio major here in about 4 months, guess what you need to be good at? Chemistry. All right, go take as much chemistry as you can. Take chemistry during intersession. Take chemistry during summer school. Take chemistry classes that they don't require. Take it. All right, take as much fundamental and introductory chemistry as you can get your hands on. Because then when you start getting into the biology, especially the biochemistry, you just go, oh, this is all chemistry. This is a piece of cake, right? Otherwise, you're dropping into a biochem class and you're trying to learn the biology and catch up on the chemistry all at the same time. And that's a tall order, right? So then here's Krebs. Again, don't be afraid of it. Throw yourself at it. And then, of course, here's ETC. Wow, really? Really? How many times have I written ETC? Literally thousands of times. There we go. Which its whole job is phosphorylation. And what do we get here? We get tons and tons of ATP. Tons of it. It's the whole point. Right? And then all I need to do is add a little off of link, which would be here, and add it off of here, and that would be CO2. That would be CO2. And there is your metabolism. I just need to hit oxygen, water. And this is a very, very big picture idea of nutrition, okay? That when you consume macromolecules, depending on what the macromolecules are, they are dropped into different locations within the cellular respiration process and converted into different substrates, which are then oxidized, the whole point being to make ATP, right? And the only true waste product is the amino group. This is not useful to us at all, right? And the CO2, the byproduct, then is returned to the plants, and they make macromolecules, and everybody's happy. Okay? Now, even for me to call that a waste, that's a little bit of a generality, because actually bacteria take that ammonia in and they fix that nitrogen to other compounds, and that makes it accessible for plants, and then plants reattach this to the amino acid, and then that's where the creation of the amino acids come from. It all comes down to plants. Plants are so incredibly useful, and most people are like, it's just plants. And really, plants are much more important than us. So there is 8B. So here's the plan. We get together on Thursday, and we'll go through the exam. And then I will ask you by Friday to decide whether you want to do the presentation Monday and Tuesday. And then we'll schedule that out for Monday and Tuesday. And then if you do it, fine, take a look at your grade and do some calculations and figure out, I'm happy with my grade, and do the exam, you'll be fine. And like, oh no, I'm at the mid B, I'd like to bump it up. I'm at the mid C, I want to bump it up. And then you decide. Okay, so you might have 3 things left, you might have 1. It's totally up to you. And then for the stoles, let's be sure and coordinate when you're going to come get those. If I have to go off of campus for any reason, let's be sure that you and I are communicating, so I can leave your stoles in the front office, because what I don't want you doing is coming up here to talk to Manro, and then Manro's not here. We can't have that happen. I do not want to waste your time.